In this video, we will discuss about the mechanism of aged CD8+T cells that cause pro-aging cognitive effect. So in order to find out this mechanism, they treated the cells, that's aged CD8+T cells with PTX that will inhibit the GPCR receptors to interact with the GPCR. So they use these three different conditions. There's young CD8+T cells with vehicle, aged CD8+T cells with vehicle, and the aged CD8+T cells with this PTX. This PTX will inhibit the GPCR receptors that are present in this aged CD8+T cells, and they did the adoptive transfer of these T cells into the mice to find out whether there is a change in this aged CD8+T cells pro-aging cognitive effect or not, using this RAWM activity. And what they are able to see is that the aged CD8+T cells alone, they are able to see more errors compared to the young CD8+T cells. And when they treat these young CD8+T cells with PTX, they are able, when they treat the aged CD8+T cells with PTX, they are able to see reductions in the error, and that is close to the young CD8+T cells, indicating that this GPCR receptors might interacting with this GPCR that are present in the mice might cause this pro-aging cognitive effect. Afterwards, they did another studies where they activate these T cells before doing the adaptive transfer into the mice, and in that case they do not see these changes. That is, aged CD8+T cells with and without PTX treatment gives the same error. Those results are in supplemental figures. Afterwards, after checking that activation of these T cells before adaptive transfer does not cause any changes even with and without PTX treatment, so they think that it's the activation of the T cells that is causing this pro-aging cognitive effect. So afterwards, what they did is they target the activations of this activated CD8+T cells using two different reagents, that is tofacitinib and anti-VLA4, and did the same studies, that is they checked the activities pro-aging cognitive effect using RAWM method. So basically they are targeting this PTX, they are using this PTX, tofacitinib and anti-VLA4 to prevent these activations of the T cells by targeting this JAK-STAT signaling pathways. So by using this tofacitinib, they are able to see that even in the aged CD8+T cells that are not treated with the PTX has shown reductions in the error, and those errors are similar to the young CD8+T cells without tofacitinib treatment. That can be seen here. That is this aged CD8+T cells and these young CD8+T cells results are similar to each other, and this aged CD8+T cells without tofacitinib results also look similar, but the p-value is 0.09. So in this case they are able to see that with tofacitinib treatment the error rates goes down further even without using PTX. And when they use this anti-VLA4, in that case also they are able to see that the error rate between aged CD8+T cells treated with anti-VLA4 and the young CD8+T cells are non-significant. Its p-value is 0.009. So this indicates that in this case also, by inhibiting the activations of the CD8+T cells or aged CD8+T cells, they can prevent the pro-aging cognitive effect. So all these results indicates that it's the activation of the CD8+T cells that is causing this pro-aging cognitive effect. and this is what they have come up from the results that are shown in this figure and the supplemental figures. So it's the activations of the aged CD8+T cells till now what they have found out. So I hope you understood the results that are presented in this figure and how they come up to this point, that activation of the CD8+T cells cause pro-aging cognitive effect. If you need to understand more about these experiments in detail, then go through the reference that has been provided in the description. That's all for now. Thank you for your time.